Welcome students. Today I will be going to deliver a lecture that's based on spoilage of fish. The main objectives cover, I will be giving you the knowledge about the general introduction of spoilage, the different types of spoilage in fish and the different methods that are used to assess the fish quality. Spoilage. Spoilage is the degradation of food such that the food becomes unfit for human consumption. Food can be spoiled by a number of means, including physical and chemical means. However, the most prevalent cause of food spoilage is microbial growth and residence in the food, which results in numerous undesirable metabolites being produced in the food that cause unwanted flavors and odors. Immediately after capture, several chemical and biological changes take place in dead fish, which can ultimately lead to reduction for human consumption. Because of spoilage. Compared to other foods, fish is unique as a substrate for microbial growth. This uniqueness stems from several important factors like the phyclotherm nature of fish, a high post-mortem pH in flesh that's typically greater than 6, the presence of non-protein nitrogen in large quantities, and the presence of trimethylamine oxide. The occurrence of spoilage in fish is manifested and perceived by end user through change in several sensory perceptions including odor, color, shape, texture and composition. Like meat, fish and other seafoods may be spoiled by autolysis, bacterial activity or chemical reactions, or most commonly by combination of these. There are various methods employed for preservation of fish such as chilling, freezing, curing, smoking, dehydration, canning, radiation preservation, etc. Fish are defined as aquatic vertebrates that use gills to obtain oxygen from water with fins consisting of a variable number of skeletal elements called fin rays. Fish is a highly perishable commodity. Fish caught at a temperature of 16 degrees centigrade remain good for hardly a day. On ice at 0 degrees centigrade, fin fish may remain good for periods of up to 14 days. And however, in contrast, beef may be stored at 1.7 degrees centigrade for several weeks. The spoilage of fish begins as soon as fish dies. In tropical conditions, fish spoils quite rapidly within a few hours of landing if not properly cooled. The spoilage rate of fish may be reduced by good handling practices and effective temperature control. In raw fish, spoilage takes place mainly due to 3 reasons, that is, enzymic action, microbial action and chemical action. Off odor and off flavor, slime gas production, discoloration and soft texture are the obvious signs of spoilage. The components involved in spoilage process are proteins, lipids, carbohydrates, nucleotides and other non-protein nitrogen compounds. The rate of spoilage is temperature dependent and lowering the temperature will reduce the rate of spoilage. Factors influencing kind and rate of spoilage. First and the foremost is the kind of fish. The various kinds of fish differ considerably in their perishability. Thus, some flat fish spoil more rapidly than round fish because they pass through a rigor mortis more rapidly. But a flat fish like halibut keeps longer because of low pH that is greater than 6 of its flesh. The condition of the fish when caught. Fish that are exhausted as a result of struggling, lack of oxygen and excessive handling spoil more rapidly than those brought in less adverse conditions. The kind and extent of contamination of the fish flesh with bacteria. The greater the load of bacteria on the fish, the more rapid the spoilage. Temperature. Chilling the fish is the most commonly used method for preventing or delaying bacterial growth and hence spoilage until the fish is used or is otherwise processed. Types of spoilage in fish. Enzymic spoilage. Autolytic spoilage is responsible for early loss of quality of fresh fish. The first enzymic change in fish muscle is the gradual hydrolysis of glycogen to lactic acid, which is known as glycolysis. Glycolysis. In the post-mortem period, glycolysis proceeds via the anaerobic pathway where the end product is lactic acid. As the lactic acid accumulates, the pH of muscle falls In fish, glycolysis will continue until the supply of glycogen is completely used up. In general, fish muscle contain relatively low amount of glycogen compared with the mammalian muscle, and final postmortem pH is consequently higher. This makes the fish meat more susceptible to microbial attack. Flavor change in fish, nucleotide degradation. The most significant enzyme deterioration is that alter flavor. The nucleotide degradation in fish muscle produce many flavor-bearing compounds. These compounds are formed by the splitting of ATP by a series of dephosphorylation and deamination reaction. Number 2 is belly bursting. Enzymic spoilage causes belly bursting in fish, especially during a period of high food intake. These fish will have a large content of digestive enzymes in the digestive tract. Such fish will degrade quickly and spoil easily soon after they are caught. Color change in fish. Color is an important factor in seafood quality. Color change in seafood is caused by enzymic or non-enzymic action such as fat oxidation or by pigments. Black-blue discoloration. Shrimp. The development of black spot in shrimp is due to the presence of an enzyme, that is polyphenol oxidase. The black spot melanin is formed by the oxidative reaction of tyrosinase on tyrosine. Lobster. Black spot occurrence during icing and frozen storage of lobster is serious problem which have resulted in significant commercial losses. Tyrosinase activity was identified in the blood with lesser amounts in other tissue. Crab. Phenolase has been implemented in blue-black coloration of crab. The enzyme is present in the blood. Various additives have been shown to prevent blue discoloration, for example, organic acids, sodium bisulfate and EDTA. The hemocyanin-induced blacking is also prominent in crab and is non-enzymic. Yellowing of fish flesh. Yellowing is associated with lipid oxidation and carbonyl amine reaction that's observed during frozen storage. Brown discoloration. Brown or yellow discoloration is caused by the reaction of the protein or the amino acids which products of lipid oxidation. Microbial spoilage. Fish spoilage is mainly due to the action of bacteria. Bacteria are present on the surface, slime, skin, gills and intestine of fish. In dead fish, bacteria begin to invade the tissues causing spoilage and production of undesirable compound. The type of bacteria on the fish is very much depending on the microbial flora of the environment. However, in the fish processing industries, two types of microorganisms are of concern. Number first is saprophytic or spoilage type bacteria. These organisms are responsible for the spoilage of the fish. The important classes of spoilage organisms found in tropical species are Pseudomonas, Flavobacteria, Acetonobacter, Aeromonas and Morozella. The spoilage bacteria are characterized by the ability to produce H2S, reduce trimethylamine oxide to trimethylamine and convert the urea to ammonia. Many volatile sulfur compounds are also produced by Pseudomonas. Formation of ammonia. Spoilage organisms convert many nitrogen compounds into off-smelling volatile bases. The non-protein compounds present in fish are good substrate for spoilage organisms. The free amino acid pool in the muscle of fish is readily utilized by typical spoilage organism by the process of deamination. This results in the formation of ammonia, which is the primary compound produced during decomposition of fresh fish. Ammonia is the major component in the total volatile nitrogen fraction, which often is used as a quality indicator for fresh fish. Formation of trimethylamine. Marine fish is characterized by the presence of an odorless compound called trimethylamine oxide. Marine flatfish and teleost contain low level of this compound, that is in the range of 0.1 to 0.5, while elasmobranchs like shark, rays, etc. and gadiotes contain very high level ranging from 1 to 1.5%. Spoilage bacteria convert this substance into foul-smelling trimethylamine. Histamine formation. Microbial spoilage of fish produce the toxin histamine in certain fishes. During spoilage, histidine is converted into histamine by bacteria. Indole production. Conversion of tryptophan to indole is another result of amino acid decomposition by bacteria. The FDA uses indole level along with sensory evaluation for measurement of shrimp decomposition. Other compounds formed during bacterial spoilage. A number of extractives are available in fish for bacterial action, such as free amino acids, sugars, peptides, creatine, as well as lipid and proteins. Chemical examination of spoiling fish muscle is shown that organoleptically the most important constituents are the volatile sulfur compounds such as hydrogen sulfide, dimethyl sulfide and methyl mercaptan. Ester of low fatty acid such as acetic, propanoic and butyric hexanoic acids are also produced. Pathogenic bacteria. The pathogenic bacteria associated with seafoods are of two types. Number 1, indigenous bacteria. They are widely distributed in the aquatic environment. These pathogens occur in minimal numbers and are not a serious problem in fresh fish. However, their growth and multiplication in seafood is a serious problem and can cause illness. For example, Clostridium botulinum, Vibrio species, Aeromonas species, etc. Number 2, non-indigenous bacteria. They occur in seafood as a result of contamination. The source includes polluted aquatic environment, sewage, excreta for animals, birds, human beings, workers handling the material as well as the surface and environment where the seafood is being processed. For example, Salmonella species, Shigella species, E. coli and Staphylococcus aureus. Number 3 is the chemical spoilage, that is the oxidation of fish lipids. Fish lipid is characterized by a high level of polyunsaturated fatty acids and hence undergo oxidative chains. With fatty fish in particular, fat oxidation gives rise to problems such as rancid flavor and odor as well as discoloration. Lipid oxidation is by two processes. Number 1 is autooxidation, that is the action of oxygen on the unsaturated fatty acids, and number 2 is lipid hydrolysis. The enzymic hydrolysis with free fatty acids. Oxidative rancidity is of great concern in fatty fish storage. Methods used to assess fish quality. Several methods are used to determine the fish quality. These can be classified into sensory and instrumental methods. The latter comprise chemical, physical and microbiological methods. Sensory evaluation of fish quality is the scientific discipline used to evoke, measure, analyze and interpret reactions to characteristics of foods as perceived through the sense of sight, smell, taste, touch and hearing. Sensory methods have the advantage of being simple, cheap and rapid. However, they can be very subjective as they are based on the assessment of individuals, their likes and dislikes. Chemical methods rely on the measurement of metabolites produced during fish storage or distribution to obtain a quantitative fish index. The most widely used chemical test is total volatile basis, that is TVB, which measures the content of trimethylamine plus dimethylamine ammonia plus other basic nitrogenous compounds associated with seafood spoilage. Other tests target the separate measurement of TMA, DMA nucleotide catabolites known as the percent K value or biogenic amine contents. Oxidative rancidity is measured by evaluating the peroxide value during the early stages or the thiobarbitoric acid-related substances (TBRS) during later stages. Fish authentication relies on the electrophoretic or DNA sequencing methods. Chemical methods are rapid, quantitative and reproducible. However, no one test is capable by itself of providing a picture of the full spectrum of changes that take place in fish and lead to its spoilage. Physical methods involve the measurement of fish, pH, texture or electrical properties. These methods are rarely used because they are either not sufficiently reliable or require calibration depending on the fish species. Microbiological examination of fish aims at evaluating hygienic quality of fish, including temperature abuse and the possible presence of pathogenic microorganisms in the fish. They consist in the measurement of total aerobic bacteria, also called as total plate count (TPC), spoilage bacteria and various pathogenic bacteria. Principles of various preservation techniques of fish. Conservation is necessary to keep the dead fish in fresh condition for quite a long time. This is achieved by employing any one of the methods like freezing, drying, salting, smoking and canning. Freezing. Freezing means removal of heat from the body. To check the enzymatic, bacterial action and putrefaction, it is preferred to store the fish under lower temperatures. The fishes are chilled in ice when they are to be stored for a few days. Ice is put inside the body cavity in large fishes. The fishes are arranged in tiers, in shells or boxes, or they may be stacked and should not be dumped in heaps in cold storage. Deep or quick freezing. When fish is intended to be stored for a long period, quick freezing is preferred, which inhibits bacterial action. During quick freezing, every part of the product comes within the range of 0 to -5 degrees centigrade. Properly frozen fish at -20 degrees centigrade retains the physical properties and nutritive values for a year or more and is almost as good as fresh fish. There are 3 ways effecting quick freezing. Number first, direct immersion of fish in the refrigerating medium. Number second, indirect contact with the refrigerant through plates. Number third, forced convection of refrigerated air directed at heat transfer surfaces. Freeze drying. This is the modified deep freezing, completely eliminating all chances of denaturation. The deep frozen fish at 20 degrees centigrade is then dried by direct sublimation of ice to water vapor without any melting into liquid water. This is achieved by exposing the frozen fish to 140 degrees centigrade in a vacuum chamber. The fish is then packed or canned in a dried condition. Any loss of flesh contents by way of leaching during melting of ice is thus avoided. The product is quite fresh, looking in appearance, flavor, color and quality. Drying. Drying involves the dehydration, that is the removal of moisture contents of fish so that the bacterial decomposition or enzymic autolysis does not occur. When moisture contents reduce up to 10%, the fishes are not spoiled provided they are stored in dry condition. Fish drying is achieved either naturally or by artificial means. Natural drying. In natural drying, the fishes are being caught and are washed and dried in the sunshine. They are suspended or laid out flat on the open ground. Artificial drying. In artificial drying, the killed fish are cleaned, gutted and their heads are being removed. They are then cut lengthwise to remove large parts of their spinal column, followed by washing and drying them mechanically. Salting. Salting is a process where the common salt, sodium chloride, is used as a preservative which penetrates the tissues, thus checks the bacterial growth and inactivates the enzymes. Dry salting. In this process, the fish is being first rubbed in salt and packed in layers in the tubs and cemented tanks. The salt is applied in between the layers of fishes in the proportion of 1:3 to 1:8 salt to fish ratio. The proportion of salt to fish varies with the fish since the oily fish require more salt. Wet salting. The cleaned fish are put in the previously prepared salt solution. It's stirred daily till it's properly picked. Mixed salting. In this process, simultaneous use of salt and brine is followed. Pit curing. In this process, the fish is treated with salt and is buried in pits lined with leaves. After 2 to 3 days, they are being removed and marketed directly. Smoking. In this method, landed fish is cleaned and brined. It's then exposed to cold or hot smoke treatment. In cold smoking, first a temperature of 38 degrees centigrade is raised from a smokeless fire. After this heating, cold smoke at a temperature below 28 degrees centigrade is allowed to circulate past the fish. In case of hot smoking, a first strong fire produces a temperature around 130 degrees centigrade. This is followed by smoking at a temperature of 40 degrees centigrade. The smoke has to be wet and dense. Canning. Canning is a method of preservation in which the spoilage can be averted by killing microorganisms through heat. It's generally well known that food carries microorganisms which cause spoilage if left unchecked. The microorganisms are to be eliminated and the entry of other is restricted. The canning process involves pre-treatment of fish, preparation of can, filling and closure of can, technique of heating the filled cans to kill microorganisms without damage to fish. Finally, cooling, cleaning and storage of the product. Methods of canning. Number first is the filling. Empty cans should be packed carefully by employing the manual labor or through mechanical device. Brine is used when fish is not salted properly as an additive for enhancing flavor. Exhausting. The air and gas from the cans should be removed before its sealing process. This can be done by using exhausting, which minimizes the strain on the can through expansion of air during heat processing. Processing. Processing is the removal of air as completely as possible. It is an important factor in steam processing. The container along with the contents of fish is heated in a retort at a temperature which is sufficient to kill the potential inactive spoilage agents without any damage of the fish inside. Irradiation treatment. Gamma radiation is considered as an innovative and interesting method to preserve chilled, stored fish and also reduce microbial population in fresh fish and fish products. Several researchers have reported increase in storage times of 1 to 3 weeks for fresh and cooked products and doubling of storage times for frozen products. Several authors have noted that irradiation doses of 2 to 7 kg can reduce important pathogens in foods such as Salmonella, Listeria and Vibrio species, including many of fish-specific pathogens like Pseudomonas and Enterobacteriaceae, which can be significantly reduced in number. The role of packaging technology. Proper packaging plays a crucial role in preservation of quality and delivery of safe, wholesome fish and seafood products to the end user. Packaging performs 3 main functions, that is, containment, protection and information. Innovative packaging technologies based on manipulating the gas exchange characteristics of packaging material to control the oxido-reduction potential have been developed and applied to preserve and extend the storage stability of fish and seafood products. The applications of vacuum packaging, controlled atmosphere or modified atmosphere packaging around fresh fish is based on the following premise, that is, some spoilage bacteria and lipid oxidation require oxygen, thus reducing the oxygen around the fish will increase storage and shelf life. Depending on the fish species and intended end use, specific combinations of oxygen, carbon dioxide and nitrogen determine the level of controlled atmosphere or modified atmosphere in practice. Vacuum packaging, CA storage and MA packaging are used in combination with refrigerated storage for preservation of fresh fish and seafood products. The combination methods must be optimized and closely evaluated to match specific requirements. With this, we conclude today's topic, and I hope you have enjoyed and have gained a lot of information regarding to the spoilage and preservation techniques of fish.